this event. I'm Aric from the Earth Environmental Sciences Department. I'm an aquatic biogeochemist, and my research focuses mostly on how microbes interact with their environment and what the consequences of that interaction are. And so I'm excited about potential interdisciplinary collaborations that work towards unique approaches to complex environmental issues. So today I'm going to talk primarily about harmful algal blooms. And harmful algal blooms develop mostly as the result of human land use change, whether that be for urban spaces or agricultural practices that lead to nutrient runoff and excess in local waterways. Harmful algal blooms have significant, expensive consequences. They can lead to fishery losses and losses of species within ecosystems as a result of toxin excretion by certain algae or the decline in available oxygen. So one of the things I'm most interested in is figuring out how do we predict bloom development and its extent, and how do we couple our understanding and predictive capacity with more efficient urban and agricultural practices that retain nutrients and fertilizer on those land use areas. And so what we're looking at here is the San Joaquin River Basin. You can see the San Joaquin River in the bottom right, draining the Sierra Nevada range and flowing northwest through the Central Valley into Stockton, into the Delta, and eventually into the San Francisco Bay and San Pablo Bay region. Along the way, the river is accumulating nutrients that flow into rivers as precipitated water flows across the surface of the Central Valley and Sierra Nevada range. And by the time this water deposits in the bay and in the coastal reaches of the state, it is high in nitrogen and phosphorus, which is prime for bloom development. So this figure is showing you in red the areal extent of algal bloom development, where the red means active photosynthetic activity and the blue means less active. And you can see that those coastal regions can develop pretty expansive bloom regions as a result of the entrainment of nutrients from those agricultural and urban areas. And so the consequences, like I said, are that these excrete toxins that can be harmful to humans and other organisms, but it also exhausts oxygen in the water column for fish. What's interested in figuring out how we predict blooms and their consequences is looking at the community from the bottom up, figuring out what bacteria and photoautotrophs are doing in response to nutrient excess, and what the consequence of that is for larger organisms as well. And so that involves a laboratory approach where I would isolate and grow these community members, microbes in the lab, and test their response to a variety of different nutrient variables, environmental conditions, and potentially some microcosm experiments where I do this in a controlled, larger scale setting in the bay or in these areas. That would all be accompanied by a time series field data set collection, where I would make use of existing USGS sampling sites in these bays and along the San Joaquin River to look at protein and gene level expression of microbes in river and in estuarine and these bay community areas to see how they are responding to nitrogen and phosphorus excess and how that response changes over time as flow conditions and nutrient availability is changing. So I'd be able to characterize the biological response from the bay into the river up to the tributaries to get a kind of source to sink understanding of what conditions are primed and we ultimately lead to bloom development. The other important component of this research is identifying the nutrient sources to the river basin, and that would focus primarily on nitrate and phosphate, the dissolved forms of nitrogen and phosphorus. The unique thing about nitrate and phosphate is they have a stabilized topic composition that would allow us to trace the source of nitrogen and phosphorus to all these different rivers and tributaries. And so on the right, you can see that there are a host of different land use types in the San Joaquin River Basin, from pink urban areas to native vegetation areas in the pale yellow. And what we would do with these stabilized topic tracers is identify the sources of nitrogen in all these different land use areas. So human agricultural waste has a unique isotopic composition different from native vegetative waste, which is different from fertilizer applied waste. Fertilizer applies nitrogen and phosphorus. And so that would allow us to identify nitrogen and phosphorus coming from unique reservoirs and how they mix in these waterways and what the ultimate effect of these is on the development of blooms and their collection in bay and coastal regions. And so with these isotopic traces, we could identify nutrient sources and get a more predictive sense for the development of blooms, but also figure out a more efficient way to effectively apply fertilizers and treat waste. If you have any other additional questions, please contact me, and thank you for your time.